Welcome to The Explainer. So today we're diving into a really cinematic journey to decode the magnificent, living skyscrapers that are literally all around us every single day. I'm talking about flowering plants, or, as botanists call them, angiosperms. We aren't just looking at standard biology here. We're exploring incredible, dynamic architecture. There's this quote from our source material that just rings so true. The wide range and the structure of higher plants will never fail to fascinate us. And honestly, it absolutely holds up. I mean, despite their massive diversity in external morphology, which is just a fancy way of saying their physical form, these biological wonders actually all share a common, elegant structural blueprint. We're talking roots, stems, leaves, flowers, and fruits. And today, we're going to learn exactly how to read that blueprint. So that begs the question, how exactly do you decode the architecture of a living machine? Well, picture this like a sweeping cinematic camera shot. We're going to ascend this entire structure from the absolute ground up. We'll start way down in the dark subterranean foundations, sweep our way up through the structural scaffolding, and finally arrive at the blooming penthouse to see how this whole beautiful system is engineered. Let's hit level 1. Plunging right below the surface, we immediately run into two very different structural strategies for anchoring these living towers. On one hand, you've got the taproot system, which is super typical of dicot plants, like mustard. It basically operates as this deep driving single main axis with smaller lateral roots branching off, kind of like a massive central support pylon. But then contrast that with the fibrous root system you'll find in monocots, like your everyday grass. In that case, the primary root doesn't stick around long. It gets replaced by this chaotic, shallow net of roots bursting right out from the base of the stem. Both get the job done and secure the plant firmly to the earth, but man, their engineering strategies are totally night and day. Now, if we zoom out just a tiny bit from the microscopic tip of those roots, we see that this foundation isn't just sitting there statically. It's actively driving through the earth in 4 distinct dynamic stages. Right at the very tip is a literal protective helmet pushing through the rough soil. That's the root cap. Just millimeters above that is the region of meristematic activity, where these tiny cells are just dividing relentlessly. Moving slightly higher, those same cells rapidly stretch out in the region of elongation, which physically rams the root deeper into the ground. And finally, we hit the region of maturation. This is where the cells differentiate, and these delicate thread-like root hairs emerge to pull in vital water and minerals from the dark earth. It's an incredible process. Okay, bursting up through the soil surface, our camera rises to the central scaffolding, the stem. This ascending axis develops directly from the plumule of a germinating seed. And what's really crucial to notice here is the rhythmic structure of it all. It's defined by nodes, which are the exact structural joints where leaves are born, and the internodes, which act as the essential spacers between those joints. The whole point of this scaffolding is to spread out branches, conduct water upward, and push the plant's solar panels directly into the absolute best possible light. Sweeping up that scaffolding, we reach those solar panels I mentioned, the leaves. The architecture here is brilliantly simple, but honestly, highly effective. You have a flexible stalk called the petiole that attaches the leaf to the stem. Think of this petiole as a dynamic mount, letting the wide green lamina or the leaf blade flutter in the wind. And actually, that fluttering isn't just for show. It actively cools the leaf and brings fresh air right to its surface, all while holding it out nice and flat to catch maximum sunlight. If we push in real close and look at that green lamina, you're going to see a complex network of veins. Now, these aren't just for transporting water and food. They provide absolutely crucial structural rigidity to the leaf blade. It's a lot like rebar inside concrete. In most dicots, these veins form the sprawling, interconnected web, which is known as reticulate venation. But if we shift our focus to most monocots, those veins run as straight, parallel highways right alongside each other, a design very fittingly called parallel venation. And how these solar panels are attached is super interesting. It perfectly illustrates phyllotoxy, which is the strategic, almost mathematical arrangement of leaves on the stem to maximize light capture and avoid shading each other out. So a plant might stagger a single leaf at each node, kind of like a spiral staircase. That's an alternate pattern, like you'd see in a sunflower. Or it might place a pair perfectly across from each other in an opposite pattern. Think of a guava plant. And in some really intricate designs like the Alstonia plant, it goes for a world pattern, where multiple leaves fan out from a single node in a complete 360-degree circle. Panning up to the very peak of our biological skyscraper, we hit the inflorescence. That's just the arrangement of flowers on the floral axis. Here, the plant literally has to decide how to cap its upward growth. In what's called a racemose pattern, the main axis just keeps growing higher and higher, with flowers blooming laterally along the sides, so it looks like a never-ending tower. But in a cymose pattern, the main axis terminates abruptly in a flower. Boom. That definitively caps its upward growth, which forces any newer flowers to branch out from below that original bloom. Right, now let's zoom all the way in and deconstruct the absolute masterpiece of this living building, the flower itself. This is the core reproductive unit. A typical flower is built of 4 distinct successive rings or whorls sitting on a slightly swollen base called the thalamus. The outer 2 rings are your accessory organs. You've got the calyx, which is usually made of those green protective sepals, and the corolla, made up of the visually striking, colorful petals. Then, tucked safely inside those protective layers are the crucial reproductive organs, the male androecium, which consists of the stamens, and right there in the very center, the female gynoecium. The spatial geometry of these floral whorls is just a marvel of precision. Seriously, when you view a flower straight on, it presents a distinct symmetry. It can be actinomorphic, meaning it has perfect radial symmetry, kind of like a wheel. Slice a mustard flower in half in any radial plane and you'll get perfect equal halves. Or it could be zygomorphic, like a pea flower, which has a strict bilateral symmetry, meaning it can only be divided into equal halves down one specific vertical line. And then, very rarely, a flower can be entirely asymmetric and just totally irregular. A really crucial detail in this floral architecture is knowing exactly where the ovary sits relative to the other whorls on that thalamus space we talked about. If the ovary sits at the absolute highest point and all the other parts attach below it, it's a hypogynous flower with a superior ovary. If the other parts attach at the rim, almost at the exact same level, it's perigynous or half-inferior. And if the thalamus grows entirely upward, completely swallowing and fusing with the ovary so the other parts actually sprout from above it, we call that an epigynous flower with an inferior ovary. Let's push our macro lens even deeper, right into the gynoecium itself. This female reproductive whorl is made of individual units called carpels. Now, when a flower has more than one carpel, they have a choice. They can either act independently and be completely free, which botanists call apocarpous, like you'd see in a classic rose. Or they can be structurally fused together into a single unit, which is called syncarpous, like in a tomato. And inside that ovary, the ovules are arranged in these very specific geometric patterns along the walls or center. That intricate architectural detail is known as placentation. Following the dramatic, microscopic transformation of fertilization, we arrive at the final harvest. The flower petals drop, the accessory organs wither away, and we're left with the fruit. And botanically speaking, a fruit is quite literally just a mature, ripened ovary. The outer wall of the ovary thickens up to become the pericarp wall of the fruit, and those carefully arranged ovules we just looked at inside harden to become the seeds, completely ready to start this entire building process all over again. Okay, so here's what's truly amazing about botany. All of this intricate three-dimensional architecture we've just visualized cinematically, it can all be collapsed into a single, elegant string of mathematical symbols. This is known as a floral formula. Let's look at the structural code for the Solanaceae family, which includes things like potatoes and tomatoes. That circle with a cross, that denotes radial actinomorphic symmetry. The combined male-female symbol means it's bisexual. K5 in brackets tells us there's a calyx of 5 fused sepals. C5 means 5 fused petals. A5 indicates 5 free stamens in the androecium, and G2 with a line underneath it beautifully maps out a gynoecium of 2 fused carpels with a superior ovary. It is literally the architectural blueprint of the plant, written on a single line. Mind-blowing, right? As we wrap up our explainer today, I really want to step back and leave you with this thought. Armed with the ability to actually read this botanical blueprint, to look at a leaf and see its veination, to look at a stem and map its nodes, to peer into a flower and deduce its symmetry and structural formula, how will your view of the natural world change tomorrow? Because next time you step outside, you won't just be looking at plants anymore. You'll be walking through a sprawling, cinematic city of perfectly engineered living machines. Keep observing, everyone, and keep learning.